Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about nucleotide and its role in formation of nucleic acids. Yes or no? So nucleotides are the monomers, simple units. When many nucleotides are attached to each other, nucleic acid will be formed. So in the same way, DNA, RNA, both of them are formed in the same way. So that is the reason why in this lecture, we will try to understand what actually a nucleotide is, what are the individual components, what are those structures. That's it. In this lecture, our aim is only to study the structures of individual components. That's it. In the next lecture, we will try to link these molecules. Do you have to understand this? So in this way, first we will study about individual structures in this particular lecture. So once we complete this, please remember to understand the next coming lectures. This lecture is the most important one, because to understand the bonds and all the things, first you need to understand the structure. And generally what many students do is they will try to skip the structures. Please remember, anyhow, once we are completing all this, we will read NCERT lines also. If you are reading NCERT lines, please remember they have very clearly they have given that so and so carbon is attached to so and so thing. Then if you don't know the structure, how you will understand those sentences? So that's why please remember, don't try to by heart these structures. Try to understand at least the logic. So what are the molecules that are linked at least? Even though you don't remember the basic structure, at least for the bond formation, you need to know the locations and all. So that's the reason why better you please try to practice these structures. And what is the advantage with this? Even in the chemistry also there is a chapter called biomolecules. At least there also you need to study the structures. Better you just study here itself, so that it will be easy for you. So the same information you can use in biology, same information you can even use in chemistry also. So that's why please remember, my suggestion is everyone should compulsorily know about these structures. Okay. So now slowly one after the other with logic we will go with these structures. Okay. Basic structure will be there, and the remaining addition, substitutions and all the things slowly we will try to understand. Clear? Yes. Now we are starting with this. So now in this lecture we need to study about the nucleotide and what are the components and what are their structures. So let us quickly once again go with the flowchart or that tree diagram. In the previous lecture we have made the same tree diagram. Nucleotide, let us quickly write this one. Nucleotide has how many components? 3 components it is having. So what are those 3 components? First one is nitrogenous base. Simply I am writing N base. That means nitrogenous base. And what is the second one? Second one is sugar, and the third one is phosphate group. Third one is a phosphate group. Clear? And now first we will focus on this nitrogenous base. And nitrogenous base, again 2 types are there. They are purines and the second one is pyrimidines. Purines and pyrimidines. Within these purines again 2 types are present. What are those? Adenine and guanine. Adenine and guanine. And what about these pyrimidines? Overall, if you are combinedly studying for DNA and RNA, total how many types of pyrimidines? 3 types of pyrimidines will come here. So what are those? Cytosine, uracil, and the last one, what the spelling? I told you to remember the spelling also. What the spelling? It is THYMINE. It is THYMINE. Okay. So in this way, totally adenine is indicated with A, guanine is indicated with G, cytosine with C, uracil with U, and thymine with T. Yes or no? This is about the nitrogenous bases. Now come for the sugars. How many types of sugars? 2 types of sugars are present. What are those? One is ribose sugar and the other one is deoxyribose sugar. One is ribose sugar and the other one is deoxyribose sugar. This is the flow chart or the tree diagram that we are actually made in the previous lecture. And now what we need to do? What is the basic structure of purine? And what are the structures of adenine, guanine? Next one, what is the basic structure of pyrimidine? And what are the structures of cytosine, uracil, thymine? Then, what is the basic structure of ribose? And then, slight modification, and what is the structure of deoxyribose? And finally, we will go with the structure of phosphate group. So, this particular lecture, you may think that this is like a chemistry lecture, because in this lecture, we are completely studying the structures of all these molecules. Clear? Now, let us quickly start with this. First, we are talking about the nitrogenous base. Nitrogenous base. Okay. All these structures, everything I will give you in the notes. And I am telling you once again, those who want to write the notes, yes, you can write it. But anyhow, we will provide the notes for each lecture separately, complete notes. So in the regular class, how we are dictating the notes, everything you will get in the app. Okay, so don't worry. But here my suggestion is, please practice these structures. So now you just try to understand how exactly I am converting from one form to other form, and what is the relation I am saying, so that when you are practicing on your own, at that time these logics will be more useful for you. Clear? Now let us go with this. Nitrogenous bases. So mainly these are nitrogen containing compounds. These are actually heterocyclic compounds. If you are clearly recollecting the biomolecules chapter, there they have given that these nitrogenous bases are heterocyclic compounds. Please remember. Generally, in the case of organic compounds, carbon will be present in those rings. But here, along with the carbon, this nitrogen, oxygen, all these are present. So that's why these are called as heterocyclic compounds. But even though they are heterocyclic, so major one is here, nitrogen is also present. Anyhow, carbon will be present. How we are expecting without carbon? Carbon will be there, but here nitrogen is also present. So that's why we are simply calling them as nitrogenous bases. If you are observing the structure, not only carbons, nitrogens, and in some cases oxygen may be present. So that's why they are called as heterocyclic compounds. Clear? That's it. Now let us come to this. These nitrogenous bases are two types. What are those two types? Purines and pyrimidines. Purines and pyrimidines. Purines and pyrimidines. Now, before directly going with structure of adenine and guanine, please try to understand NCERT wording given in biomolecules. What is the word they have given in biomolecules? Please observe carefully. In the purines and in the purines, we are talking about adenine and guanine, right? So generally what we say, adenine and guanine are the purines. This is the statement. Yes or no? Adenine and guanine are purines. Correct, no problem. I am not saying that is wrong. That is a correct statement. But what is more technically correct? Observe this statement carefully. Because when we are giving this particular statement in examination, many students are making mistakes. So that's what the statement I am saying. Adenine and guanine are substituted. Please observe this word, substituted purines. Now tell me whether it is true or false. If they are saying that adenine and guanine are purines, yes, no problem. Adenine, guanine are purines, and cytosine, uracil, thymine are pyrimidines. We are generally studying, so no problem. But if they are saying that adenine and guanine are substituted purines, true or false? Tell me this. These are the very important things. These wordings were clearly given in biomolecules chapter. They have not given here, but in biomolecules they gave in class 11th. So please observe. It is a correct one. Same way here also. Cytosine, uracil, thymine are substituted pyrimidines. Substituted pyrimidines. Now you need to understand the logic. Why they are using the word substituted? Why they are using the word substituted? Because purine is having a specific structure. That's why generally what we say, so adenine and guanine have purine basic ring. So basically they are having the purine ring, and on that small changes are there. So that's why they are considered as a purines. So technically these are the substituted purines. That means these two are having basic purine ring. They are having basically purine ring containing molecules. That's why they are called purines. And here these are basically having pyrimidine ring, and then it is substituted. So that's why they are considered as pyrimidines. Clear? So that's why adenine and guanine are substituted purines, and cytosine, uracil, and thymine are substituted pyrimidines. This is more technically correct, but still generally you can say that adenine and guanine are purines, cytosine, uracil, and thymine are pyrimidines. Correct, no problem. So please understand this word. Why they are using this? Logic also you please try to understand. Then you can easily understand what is the meaning, why they are giving, and everything. So now what is the point that we understood? First let us focus on purines. What is the meaning of this particular one? There is basically a particular purine structure. On this purine structure, some groups are substituted. This is what the meaning is. Same way, now we will study about this particular one. So first we will study about the purine. I will write the structure of purine at the middle here. What is actually a purine structure? Purine actually has two rings in it. So this is one ring, okay, and the other ring is like this. Clear? And where the double bonds are present? Here, 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 and here. Understood or not? So this complete structure is called as purine structure. Please understand this. So technically in this purine structure, this is one ring and this is another ring. So two rings are fused. Two rings are fused to form the purine structure. This is basically a purine structure. And what are the names of this one? This first ring itself is called as a pyrimidine ring. So please observe this. Many students are getting confused here. You are talking about purine and here you are saying pyrimidine. So please observe this. So here if you are talking only this first ring, left side ring, that ring itself is called a pyrimidine ring. So if you know this purine structure, you will automatically know what is pyrimidine structure. So that's why these are all very simple. Some students are unnecessarily getting confused, they are overthinking and they are thinking that you cannot remember. Very simple thing. This first ring itself is pyrimidine. So that's the same thing we will use for pyrimidines also. Okay, fine. Now what is this right side ring? This right side ring is called as imidazole ring. Imidazole ring. Understood or not? So in this way, here it is one heterocyclic ring. This is also one heterocyclic ring. These two are fused. So one is pyrimidine ring, other one is imidazole ring. And these two rings are fused so that now they are forming this structure called as a purine. So purine is having heterocyclic rings in it. Clear? Over. Now numbering. What is this numbering here? Please observe this carefully. Numbering will start from this. This nitrogen will get the first position. And here is the carbon, right? So wherever you are not mentioning, please remember these positions are carbon positions. These are all carbon positions. Clear? Yes. So this is the second, third, fourth, fifth, and sixth. With this first ring is completed. Okay. And in the second ring, we are coming like this 7, 8, and 9. So if you are clearly observing this, in the first ring, we are going in anti-clockwise direction. And for the second ring, we are coming in clockwise direction. Please observe this. So in this way, total 9 are present here. Out of these 9, what are the positions of nitrogen? What are the positions at which nitrogen is present in the basic ring? Within this basic ring, what are the positions of nitrogen? First position, nitrogen is present. Third position, nitrogen is present. Seventh position, nitrogen is present. Ninth position, nitrogen is present. So within the basic structure of the purine, total 4 nitrogens are present at the positions 1, 3, 7 and 9. That's it. This is the basic purine ring structure. Clear? And here what you need to understand is bonds and satisfaction. So what is the valency of these things? Carbon will form how many bonds? 4 bonds. Nitrogen will form how many bonds? 3 bonds. And oxygen will form how many bonds? 2 bonds. And hydrogen will form how many bonds? 1 bond. Please observe this. Based on this, you need to satisfy at each position, okay? And now we will understand this particular point. And based on this, so here this is the number, right? So this number should be maintained, okay? If at any location, if these many bonds are not present, that means by default, automatically their hydrogen will be present. Is it clear? First we will start with first position. First position is nitrogen. How many bonds should be there? 3. What are the bonds? 1, 2, 3 bonds. Yes or no? This is the sigma bond, this is the sigma bond, this is the pi bond. 3 bonds are there. Fine. Clear. Next one. Come to the second one. Second one is having carbon. Carbon actually should have how many? 4. First bond, second, third bond. Only 3 bonds were mentioned. What about the fourth bond? So if it is not mentioned, means by default here hydrogen will be present. So fourth bond will be with hydrogen. Please understand these things. Next one. Coming to the third position. Nitrogen. 1, 2, 3. Enough. Clear. And fourth one. Carbon it is. 1, 2, 3, 4 bonds are present. So it is satisfied. Fifth one. Carbon. 1, 2, 3 and 4. Satisfied. Clear. Sixth one. It is carbon. 1, 2, 3. Only 3 are mentioned. Then what about the fourth one? It will be with hydrogen. That's it. Seventh one. Nitrogen. 1, 2, 3 bonds are present. Satisfied. Eighth position carbon. 1, 2, 3 are there. Then what about the fourth one? It will be with hydrogen. N should have 3 bonds, but here only 2 were mentioned. Then what about the third one? It will be with hydrogen. So in this way, wherever this valency is not satisfied, there it will be with hydrogen. This is the default condition you need to understand. I hope already you know about all these basic things. Already you might have studied all these things in chemistry 11th class, right? This is what about the basic structure. Clear? And here, now let us come to the next one. So this is the basic structure of the purine. So those molecules which are having this basic purine structure are called as purines. That's it, simply. Now we will talk about the adenine. Now we will talk about the adenine. It is indicated with A. Okay. When you are drawing the structure of the adenine, as it is purine structure, you just copy here. As it is a purine structure, you please copy without any change. Without any change, even without change in double bonds, as it is, you please copy this. Clear? Everything as it is you have written here. And now you need to focus on the position that is called sixth position. Is this sixth position or not? Yes, sixth position. Now what happens in the case of adenine? In the basic purine ring at the sixth position, amino group is present. This is the change. Now this is called substituted purine, and this is adenine. Understood or not? So this is the purine. It is substituted with, here originally what should be there? Here carbon means 4 bonds should be there, and the fourth one with the hydrogen, right? Instead of hydrogen, now NH3 is present. So it is substituted. NH3 is substituting hydrogen. So that's why it is substituted purine. This is the reason why in the NCERT they have given that adenine and guanine are substituted purines. Clear? Now, if you are technically writing the name, at the sixth position, amino is there. So 6 amino, yes or no? 6 amino purine. Okay, but please remember here, for the N already here one bond is there. Here NH3 will not come, and here only NH2 will come. Please understand this amino group. Okay, so nitrogen will have valency of 3. Already one bond is with this, and only 2 hydrogens can be attached. So NH2. So it is not NH3, NH2 group will be attached. So that's why it is called as 6 amino purine. Clear? So adenine is 6 amino purine. It is indicated with A, and this is the structure of adenine. Completed. Now we will talk about the guanine. For the guanine also, basic purine ring as it is you please write, but this time don't draw the double bonds because there is a small adjustment that I will tell you. So here what we need to do? Draw the basic structure of purine without double bonds. There is a small adjustment here, so that's why don't draw the double bonds. And here what happens? Positions. Focus on these positions. This position and this position. This position and this position. What actually happens in the case of guanine here? At this position, NH2 is present. That means amino group is present. And at the sixth position, what happens? Double bond O is present. This is the small change. Now, according to this, now you just keep the double bond. Now tell me, for between the 2 and 3, can you keep double bond? Yes, you can keep double bond, no problem. Between 4 and 5, yes, you can keep the double bond. Between 7 and 8, yes, you can keep the double bond. But the problem will come for 1 and 6. Between 1 and 6, now you just observe carefully. Can you keep double bond here? No, because for this carbon, 1, 2, 3, 4, already 4 bonds are there. So you cannot keep double bond. So don't keep double bond. If you are not keeping double bond, what happens? For this nitrogen, 1, 2 bonds are there. So by default, third one will be with hydrogen. That's it. So simply leave it like this. If you want to write, you can write hydrogen. Even though you are not writing hydrogen, no problem. This will be like this. This is the structure of guanine. Then how the name will be written? So what are the positions here? This is the sixth position, this is the second position. Please remember these names. These names are not IUPAC names. These are not IUPAC or IUPAC names. So you don't need to worry about first you need to write this first, then you need to write. Don't worry. Randomly you can write this. First I am writing this one. It is a 2 amino. At the second position it is amino. And at the sixth position it is oxy. Double bond over at oxy. So 2 amino, 6 oxy. And what is this basic structure? Purine. So purine. Understood? So adenine is 6 amino purine. Guanine is 2 amino, 6 oxypurine. That's it. Very simple. This is what the structure is. So for all these things, first you need to know about this basic structure. Understood or not? Please remember this. And with this, purine structures are completed. Now we are going for pyrimidine structures. In the pyrimidine structures also, please observe this. This first ring itself is called pyrimidine. And in the pyrimidine, you will study about cytosine, uracil, thymine. For all the three, their numbering is different, but locations, remember these two locations, there only every time substitutions will come. Very simple structures, okay? Please remember this. With this, under the purines, we have completed adenine and guanine. Clear. Now we are going for pyrimidine. Pyrimidine means first ring, right? So please remember that first ring, pyrimidine. Pyrimidines. Pyrimidines. In the case of pyrimidines, what happens? There is a pyrimidine ring and that will be substituted. So cytosine, uracil, thymine, these three are called as substituted pyrimidines. Now we will see the pyrimidine ring. First ring itself is pyrimidine, right? Remember this. As it is, same ring we will draw here. Here we are drawing this same ring. As it is, without any change. This is a single line only, like this. Okay. Here is the double one, here is the double one, here is the double one. As it is, if you want to check it, check it. As it is, only the first ring you please draw this. This ring itself is called a pyrimidine ring. Over. Structure is completed. But what is important here? Numbering is important. Here, as part of the purine, here what is the numbering? You have started number one with this nitrogen and you have gone in anti-clockwise direction. But in this case, it is not like this. In this case, here number one will start with this nitrogen. Please remember these things. These numberings are also a little bit important. Though numbering is not important in biology, but definitely it is important in chemistry. Please remember this. Number one will come here, and now this time you need to go in clockwise. So this is the number 2, number 3, number 4, number 5, number 6. Understood or not? And in this case, what happens? For all the three, substitutions are at these two positions. That means second position and fourth position. Only these two are involved. Okay. In the case of thymine, fifth is also coming, but these two are compulsory for all the three. So now we will go with those structures. First one, cytosine. For these cases, what we will do is, first we don't draw the double bonds. First we will draw the basic one. Then we will add the groups. Based on the groups, we need to adjust double bonds, right? So then we will draw the double bonds. Now let us see. First we will draw the structures without double bond. Okay. This is the position. Please remember these two are the positions that will be affected every time, right? So please remember this. Now already in the class 10th or somewhere you studied that in the double helical DNA structure, if in one strand adenine is present, adenine will pair with thymine of opposite strand. Yes or no? If guanine is present in one strand, guanine will pair with cytosine of opposite strand. Here I am trying to tell you one logic for structure, right? So please remember. So now, just now we have studied about the structure of guanine. In the structure of guanine, forget about this. See these two positions. In the guanine also these two positions. In the cytosine also the same two positions. But here the numbering is 2 and 6. But here the numbering will be 2 and 4. 2 and 4 will be the numbering. Simple logic. Whatever the groups you have written for the guanine, same two groups you need to write for cytosine also, but locations are changed. Understood or not? Same two groups, change the location. That means in the case of cytosine, what happens? Here double bond O, here NH2 will come. So simply just write here double bond O, here NH2. Simple. That's it. This is the structure of cytosine. But you need to add double bond. Can you keep double bond here? This time you cannot keep double bond because this carbon has 1, 2, 3, 4 bonds. So leave it. Leave it means this nitrogen will automatically get third one with hydrogen. That's it. Simple logic. And here double bond as it is. That's it. That is the structure of cytosine. Very simple structures. Very simple structures. Okay. Don't get complicated. So observe carefully. You will understand this. Now how we will write this? At the second position oxy, fourth position amino. You can write 2 oxy 4 amino, or you can write 4 amino 2 oxy. Anything you can write, no problem. So now this will be 4 amino 2 oxy pyrimidine. That's it. This is the chemical name. 4 amino 2 oxy pyrimidine is the cytosine. That's it. Very simple structures. With this, cytosine is completed. And now we will go with the uracil. Uracil structure is very simple. So now we will draw the basic structure without double bonds. And here also the same two positions. But this time what happens is, at both the locations it is double bond O. At both the locations it is double bond O. Please observe carefully. At both the locations double bond O means, can you keep double bond here? No, you cannot keep. So this nitrogen will get hydrogen. Can you keep double bond here? No, you cannot keep, because already this carbon has 4 bonds. So you cannot keep double bond. So this nitrogen also getting one more hydrogen. But can you keep hydrogen, sorry, double bond here? Yes, you can keep it. So this is what? So slight change. So what is the name of this one? This is the second and this is the fourth position. 2,4-dioxy. 2,4-dioxy, right? 2 oxygen. Here is one oxygen, there is one oxygen. 2,4. So 2,4-dioxypyrimidine. 2,4-dioxypyrimidine. That's it. Very simple. This is the structure of uracil. Who said that this is difficult? Very, very simple thing, right? Over. Now the thymine. Now you need to be little bit more careful, because adenine present in both DNA and RNA. Guanine present in both DNA and RNA. At least this information already you studied in biomolecule, right? Cytosine present in both DNA and RNA. So these three are present in both DNA and RNA. But uracil and thymine. So uracil is present only in RNA. Very good. It is present only in RNA. But what about the thymine? Thymine is present only in DNA. That means here you are observing some difference. So this is present in RNA, and whatever we are writing now, that will be present in DNA. But what is the structural difference here? These points, this structure based points were discussed later on in the NCERT. In the same chapter, molecular basis of inheritance, later on after a few lectures, we will study about that content. There they have mentioned this point. So please observe carefully here. What is the structure of thymine? As it is uracil, if wherever double bond is there, keep that one also. As it is uracil, you please draw this. As it is uracil. This is the uracil. Here is the double bond, here is the double bond, here is the double bond. Double bond O, double bond O. This is the normal double bond between two carbons. As it is structure of uracil. Exactly as it is. But there is a small change. What is that change? This time you need to add one more. What is this position? Number 5 carbon. This is the 4, this is the 2, right? As it is uracil, and here you need to add the methyl group, CH3 group. Methyl group you need to add at the 5th position. Understood or not? So as it is uracil, but it is having one extra. What is that extra? Methyl group. At which carbon? 5th carbon. So now how you are writing this? 5-methyl. At the 5th position, methyl group is there. And what is this one? 2,4-dioxy. 2,4-dioxypyrimidine. 2,4-dioxypyrimidine. This is the structure of thymine. 5-methyl 2,4-dioxypyrimidine. Now you can also write this in a different way. What is this different way? 5-methyl you can write as it is. 5-methyl you can write as it is. What is this 2,4-dioxypyrimidine? It is nothing but uracil. So you can write this as uracil. Understood or not? So thymine, chemically called as a 5-methyl 2,4-dioxypyrimidine, or simply it is also called as 5-methyl uracil, very clearly mentioned in the NCERT. And this question was asked once in NEET examination in the chemistry part. Very simple question. They have asked that thymine is 2-methyl uracil, 3-methyl uracil, 4-methyl uracil, 5-methyl uracil. Like that they have given some options. What is the answer? It is 5-methyl uracil. Very simple. With this all the nucleotides are completed. Sorry, nitrogenous bases are completed. Nitrogenous bases are two types. They are purines and pyrimidines. In the purines, this is the basic ring structure, purine structure. Adenine is 6-aminopurine, it is indicated with A. Guanine is 2-amino-6-oxypurine and it is indicated with G. Over. Information is very simple. And regarding the pyrimidine, it is having this pyrimidine ring. And all these three are substituted pyrimidines. And here the cytosine is 4-amino-2-oxypyrimidine and it is indicated with C. And uracil is 2,4-dioxypyrimidine and it is indicated with U. Thymine is 5-methyl 2,4-dioxypyrimidine and it is indicated with T. That's it. These are the structures. So please practice the structures for the next lecture. Please remember. Okay. So this is what about the nitrogenous base. That part is completed. And next we are going for the sugar. Next one we are studying is sugar. Please remember this. What are these sugars? There are two types of sugars. One is called as ribose sugar. Where this ribose is present? Ribose is present in RNA only, whereas other one is called as deoxy. Simply you are saying deoxy, but you need to mention, better you please mention the position of deoxy also. Understood or not? At the second carbon position, this deoxy is there. So T 2-deoxy ribose sugar is present. Okay, this is what. And what I will do is, regarding the ribose, I will write on this side. For 2-deoxyribose, I will write on this side, so that you can easily understand this. If you are observing the content of ribose, you can easily understand that, because it is just a simple change. Okay, let us quickly go through this. What is this ribose sugar? Already in the biomolecules you studied about carbohydrates. In the carbohydrates you studied monosaccharides, disaccharide, trisaccharide, and then like the many, and finally polysaccharide. So generally you are classifying as monosaccharides, oligosaccharides, and finally polysaccharides. Yes or no? Under the monosaccharides, 3 carbon containing trioses, 4 carbon containing tetroses, 5 carbon containing pentoses, 6 carbon containing hexoses, 7 carbon containing heptoses you studied. Now come to the 5 carbon. Under the 5 carbon containing pentoses, ribose sugar, deoxyribose sugar are some examples. Yes or no? So now come to this. So this ribose is having how many carbons? 5 carbons. It is a monosaccharide. What is the basic formula for the monosaccharide? It is CnH2ON times. Yes or no? So generally what we are saying, carbohydrates are hydrates of carbon, you are saying, but originally they are not hydrates of carbon. Please remember, for the molecular formula you can get this derivation, but originally they are not hydrates of carbon. Please remember, they are polyhydroxy aldehydes are ketones, but they are not hydrates of carbon. Please remember. So this is the general formula. Same general formula you can also write as Cn H2n, H2 into n, 2n, and O n. You can write like this or you can write like this. What is that? N indicates number of carbons. So now, will this ribose follow the basic formula of monosaccharide or not? Yes, it is following. So what is the N value here? 5. N value is 5 means Cn means C5, H 2n, 2 into 5 is what? 10. So H10, ON, O5. So this is the molecular formula for the ribose. So it is following the general formula of monosaccharide. So Cn, H2n and ON. So it is C5, H10, O5. That's it. So 5 carbons are present. What are these carbons? Carbon 1, 2, 3, 4 and 5 carbons. These are the 5 carbons. Yes or no? And ribose is, please remember, for all these sugars at the last carbon CH2OH is fixed. So please keep first one. I need to give the numbering, right? This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Clear? Nice. And what about the ribose? In the sugars itself, in the monosaccharides you studied that some type of sugars are having aldehyde group and some type of sugars are having ketone group. Yes or no? Aldehyde group, definitely first carbon. So ketone group, so generally it is present in the second carbon. Clear? But what is this ribose? Ribose is 5 carbon containing. So that's why it is pentose sugar. Ribose is a pentose sugar. Penta means what? 5. Penta is 5. Already you studied all these. That's why quickly I am going through this information. So ribose is a pentose sugar. 5 carbon containing sugar. That's why pentose sugar. Good. But does it contain aldehyde group or ketone group? This is very important. This is aldose sugar. What is aldose and ketose? The sugar having aldehyde group is called aldose sugar. The sugar having ketone group is called ketose sugar, right? Now tell me, this ribose is aldose or ketose? Does it contain aldehyde group or ketone group? That's what my question is. What is present in this? Aldehyde group is present, so it will be CHO. Aldehyde group, so definitely this will be the first carbon. So CHO. So first one, last one we have completed. Now remaining 2, 3 and 4th carbon positions. And at these positions what happens is, on one side OH is present, on the other side H will be present. I hope already you studied all these structures in the biomolecules. So that's why, anyhow, we have discussed all these things with clarity, so you don't need to worry. So here, now let us directly go with the answer. In the case of ribose, what happens? In the case of ribose structure, please remember, so if you are changing this again, a new molecule will come. So this particular one is only for ribose. In the case of ribose, at second, third, fourth, at all the 3 positions, On the right side, OH is present. Left side, H is present. Understood or not? And this is what the structure of ribose. And this is ribose sugar. This is the ribose sugar. And here, please observe, slowly we are going into the other content. And this content already I have discussed in the biomolecules. So that's why we are continuing here. And what type of sugar is this? Is it capital D or capital L? Now tell me the answer. So isomers is a very important concept in the chemistry, right? Capital D, capital L. Capital D, capital L is one type of isomerism. Small d, small l is another type of isomerism. Small d is also called plus, small l is called minus. Okay. And next one is R and S type of isomerism. Yes or no? Not only this, positional isomers are present. Different types of isomers are there. Now my question is this one. Capital D, capital. Don't confuse between capital D, capital L and small d, small l. These two are completely different. Yes or no? Small d, small l is based on rotation of plane polarized light. So you are taking this compound in a tube and then you are passing the plane polarized light. If it is turning towards the right side, it will be D. D means what? Small d means dextrorotatory. And this one is levorotatory. Understood or not? So dextrorotatory means turning that light towards the right side. And levorotatory means turning it towards the left side. So turning towards the right side, dextro, small d, indicated as plus. Turning towards the left side, levorotatory, indicated as minus. This is completely different. Whereas capital D, capital L, that is completely different. Capital D, capital L, here we are giving based on the position of OH group on the last chiral carbon. Chiral carbon is also called asymmetric carbon. Please remember this. Already we have discussed this information in biomolecules, so I am not discussing once again here. Simply tell me what is the definition of asymmetric, or chiral. What is the meaning of asymmetry? There is no symmetry. What is symmetry? If the same is present on both the sides, that is called as symmetry. Then what is asymmetry? With respect to carbon, tell me what is asymmetric carbon? Carbon having, anyhow it is having 4 different bonds. On 4 bonds, 4 different groups should be there. If at least 2 are matching, it is not asymmetric. Please remember this. What is asymmetric carbon? A carbon that is attached to 4 different groups. 4 different groups is called asymmetric. So in this particular structure, if you see, first carbon C is having double bond with O. Double bond with O. So it is not asymmetric. That will come under symmetry one. So leave it. So this one 4 different groups. This one 4 different. This one 4 different group. But this carbon with hydrogen, 2 different bonds with again same hydrogen. So this is also not asymmetric. So only these 3 are called asymmetric carbons or chiral carbons. So this information is important. And how you will decide this capital D, capital for this? If OH group is present on right side, for which one? For the last chiral, whatever is the number of chirals are there, on the last chiral carbon, last asymmetric carbon, if OH group is present on right side, this is capital D ribose. Please observe this. Understood or not? Observe carefully. On the last chiral carbon, if OH is present on the right side, it is capital D. If OH is present on the left side, suppose this OH is here, H is there, then it will become capital L. Simple. Understood or not? So this is what. So in this way, if they are talking about specific isomer, is it capital D ribose or small capital L ribose? It will be capital D ribose. Please observe this carefully. This is how we are drawing the structure of ribose. But please remember, we are talking this structure inside our cells because DNA and all are present inside cells. Inside the cells means whether it is DNA or RNA, whatever it is surrounded by the fluid. What is that fluid? Observe this carefully. Suppose you are talking about a cell and this is the nucleus. DNA is present inside the nucleus. And within this, there is a fluid. Water is present, right? And RNA, here also some types of RNAs are present and some types of RNAs are coming into cytoplasm. And cytoplasm means here also water is present. Please observe this. So this DNA and RNA are surrounded by water. So now you need to talk about another concept here. So since they are surrounded by water, now come back to this. This is called as a linear form. This is called as a linear structure of ribose. But when you will see this you will not see when it is in the liquid. If it is in the solidified form, generally you may see this one. But when it is in the water, when it is in the water. You cannot see linear form, it will become circularized. It will become ring form, circular form. What is that ring form? Now let us see this. For the same ribose, please remember this side I am writing for the ribose, because on the other side I will write for deoxyribose. For the ribose only we are writing here, D-ribose only. Previous one is linear, now we need to make the ring form. In the ring form, what you need to do? First carbon, second, third, fourth, fifth carbon, CH2OH. Write this. In first carbon don't write, but second carbon OH write, third carbon OH write, and here hydrogen, hydrogen, hydrogen. All these things you just write. In the previous structure what we did? As it is I have written. Here you wrote CHO, and here you wrote for the fourth one on the right side OH. And here what happens? And here OH is there, and here it is like this, right? Now these two will interact here. These two will interact, so that there is a small rearrangement. What is that rearrangement? Observe very carefully. Okay, I will use a different color so you can understand this. What is this rearrangement? Observe carefully. Here this is the O, and it is like this, ring form. So that here again rearrangement will occur. And in this rearrangement, now you will get two options. What are the two options? In one option, you will get like this. Yes or no? And in the other option, what you will get? Reverse. In the other option, here H will be there, here OH will be present. Understood or not? So in this way, this linear form, in the solution, it is converted into this ring form. Understood or not? And when this ring is formed, now on the carbonyl carbon, please remember, aldehyde group, ketone group, these are called as carbonyl carbons. This carbon is called carbonyl carbon. On the carbonyl carbon, on which side this OH is present? Now in this diagram what happens? OH is present on left side. OH is present on left side. So this is called beta form. Please remember here. There are two forms. One is alpha form, other one is beta form. When you will say alpha form, if OH is present on right side, H is present on left side. Suppose it is like this. It is like this, then it will be alpha form. But originally, please remember this point. Why we are talking about this? Originally, inside the nucleic acid, whether DNA, RNA, whatever it is, what is the form that is present? It is always beta capital D. Why it is beta? On the carbonyl carbon, OH is present on the left side. That's why it is beta. And why it is capital D? Because on the last chiral carbon, OH is originally present on right side. So here it is right side, so that's why it is capital D. Here it is left side, that's why it is beta form. So which of the following type of sugar is present in RNA? I will give you four options. Alpha, capital L. Suppose let us write like this. Alpha capital L ribose is one option. Second one, alpha capital D ribose is another option. Third one is beta capital L ribose is another option. And the last option will be beta capital D ribose. Please remember, RNA is having ribose, fine, very good. Ribose is there every time. But what type of ribose is exactly present? This is the important. What type of ribose is present exactly? Capital D and beta form. So beta capital D ribose. This is the ribose that is present in RNA, Please understand this. Understood or not? So that's why we are writing about this one. Nice. Next one. Come back to this. So that's why the type of ribose that is present is beta form. It is not alpha form. So beta capital D ribose. Clear? Okay. And this alpha and beta are called as anomers. 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 In the chemistry, you will get more details regarding this, because in the biology generally we are not studying this. So please understand this. So this is the beta capital D ribose. Clear? This is the ring form. But and this one and the previous one, so we are writing generally in a linear form like this, and these are called as Fisher projections. Fisher projections, all these names you will study in chemistry, but these are interlinked concepts, so you need in biology and also you need in chemistry. Okay, don't get confused. It is compulsory information. You should compulsorily remember this. This is not out of NCERT. This is from the NCERT only. You need to remember this, and these are called as Fisher structures. But Generally, when we are writing on the paper, it is difficult to make this type of structures. Please remember this. So that is the reason why we are going for Haworth's projections. Haworth's projections. What is this Haworth projection? So what we do here is we will use one ring here. And for the representation of ribose sugar, what type of ring we are using? Already we have studied this in biomolecules. What type of ring we are using? We are using a ring like this. Please remember, whenever we are drawing the structure of ribose from here onwards, you need to always use this type of ring only. Don't draw like previous one, because that will be a little bit difficult for us. So this type of rings only we are using from next lecture onwards. That's why I am telling you all these points. Observe carefully. This is what the ring is. What is this ring is called as? This ring is called as a furon ring. This ring is called as furon ring. And what is the important feature of this furon ring? In this furon ring, now there are two questions are there. What are the questions? How many carbons are present in the ring? In the basic structure of the ring, how many carbons are present? And in the basic structure of the ring, how many members are present? Questions are very clear. Carbons means only carbons you need to count. But members means carbon, nitrogen, oxygen, whatever they are present. Everything you need to count it. This is the furon ring. Now let us count the carbons. Carbon, this is the carbon, this is a carbon, this is carbon, and this is carbon. In the basic structure of this, furon, how many carbons are present? 1, 2, 3, 4 are there. So only 4 carbons are present in the basic structure of this furon ring. But how many members are present? 1, 2, 3, 4, and this also you need to count. If you are talking about members, you need to count all the elements. So 5 members. So furon ring is 4 carbon 5 membered ring. Good. And for the representation of ribose, we are using this particular ring. Please remember this. This is the ring that we are using. And with the use of this particular ring, there is a small problem. What is that problem? Please observe carefully. In the original structure of ribose, how many carbons are present? 1, 2, 3, 4, and 5. How many carbons are present? 5 carbons are present in the basic structure of ribose. But in the basic structure of furon ring, how many carbons are there? Only 4 are there. Then where you will represent the fifth one? So this is the important point you need to understand. So out of the 5, please observe, out of the 5, you can represent only 4. Then what about the fifth one? Now I will give you two options. Try to understand my question first. First option. What is the first option? Out of the 5 carbons, how exactly you are representing this? Will you give carbon number 1, carbon number 2, carbon number 3, carbon number 4? So fifth one is projected outside. This is the option 1. What is the second option? Second option is first one is projected away. First carbon is projected, so that this will be second carbon, this will be third carbon, this will be fourth carbon, this will be fifth carbon. I am giving you two options. Out of the 5 carbons, we can represent only 4 carbons in this particular ring. So which option is correct? Is it the first option like this, or the second option like this is correct? Which one is correct? This also you need to understand. Then only you can draw these structures without any mistake. So please observe this. Okay. So this is what. Observe carefully. First option is correct. Please remember, in that ribose sugar, out of the 5 carbons, first 4 carbons are represented within this furon ring, so that fifth one is projected away. Fifth one is projected away from this. Like this we are representing. Is this clear? So this is the correct representation. So please know which one is the correct, which one is the wrong, because these are all very simple basic things. But generally students are making mistake in simple things only. They are trying to understand, remember all the complicated things, but they are forgetting these simple things. So please don't make mistake. So this is how we are representing. So first carbon, second, third, fourth, and fifth one. Then how you will represent this information? So actually it is present like a linear like this. And here we are having for every carbon left, right side, left side, right side, left side, right side, left side, like this we are having. But there how you will represent? So this information is very clear. So here what we are doing is, here this information, see here, first carbon is having directly O connection with fourth carbon. Already this is represented here. First carbon, O connection with fourth carbon, completed. Now for remaining other things, how we are representing, this information is very important, observe carefully. And here what we are doing is, for the first carbon, right side H, left side OH, please remember. So when you are converting this into the ring, observe carefully here, this is the carbon suppose. Right side information should be written below the plane, suppose this is the plane, you should write it below the plane. And the left side one you should write above the plane. So this one slightly turn like this, you will get the answer. For this carbon also turn like this, turn like this, turn like this, you will get the answer. So here for the first carbon, right side H, left side OH. So how you will write this? Right side H is below, left side OH is above. Completed. Next one. For the second carbon, right side OH, left side H. Right side OH, left side H. Completed. Next one. Third carbon, right side OH, left side H. Right side OH is below, left side H is above. Completed. And for the fourth carbon, we don't need to worry. Fourth carbon, already this side is in the bond. So this we are simply writing below the plane. This generally we are representing below the plane because above we are representing the fifth carbon. So that is for the fifth carbon. For the fifth carbon, simply you just write CH2OH, no problem. That's it. This is how we are generally writing the ribose sugar. This is how generally we are writing the ribose sugar. And what is this ribose sugar? This is beta capital D ribose. So please remember, from the next lecture onwards, whenever I am asking you to write ribose means, this is the structure you need to write. Please remember this. This is how you are representing. Clear? And here sometimes what happens is, see, in this structure deliberately we have mentioned hydrogens. Even though you have not mentioned hydrogen, it is okay. But what is the mistake done by the students? So the same thing I am trying to write here without hydrogens. OH, 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 and this is CH2OH. Observe this. OH, 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 CH2OH here. Clear? Yes or no? So this is the first carbon, second carbon, third carbon, fourth carbon, this is the fifth carbon. Clear? And now what is the mistake done by not just students, even many teachers are also teaching it in a wrong way. So please observe this carefully. So the thing is, if you don't want to write hydrogen, suppose you don't want to write hydrogen. If you don't want to write hydrogen, what you need to do? Simply leave it like this. Simply leave it like this. But what is the mistake done by the many students and teachers is, they are making a line, and then they are leaving like this. This is a main blunder mistake. So please understand this. If you are making a line and leaving, suppose there is a carbon here and you are making a line and leaving means what? What is the default condition in the chemistry? By default carbon will come and that will be satisfied with hydrogens. Means what? Methyl group will come. Suppose you are making a line and leaving means what? Here you are writing CH3 methyl group, then it is not ribose sugar. If you are making a line means write hydrogen. If you don't want to make a line, leave it. If you don't want to represent hydrogen, don't draw any line, leave it simply. Suppose you are drawing this line and leaving means, again it will be a mistake. If you are drawing line means, write hydrogen. If you don't want to draw hydrogen, don't draw any line also, simply keep it like this. This is the one thing that you need to understand. So in this way, you need to understand this. So that is the reason why, anyhow, once we are going through NCERT diagram, I will show NCERT diagram. In the NCERT diagram, this type of mistake was given. Actually, hydrogen should be represented. They made a line and they left it like this. If you are drawing a line and leaving means, please remember CH3 methyl group will come there and it will completely change our structure. Please understand this. We want this type of structure. So that's why please learn each and every point technically so that there will not be any mistake. Okay. So this is what about the ribose sugar. That's it. Simply it is about the structure. So it is a 5 carbon containing sugar. That's why pentose sugar, and it is having aldehyde group. That's why it is called aldose sugar, and its chemical formula is C5H10O5. It is C5H10O5. That's it. Clear. With this, ribose sugar is completed, and this ribose sugar is present only in RNA, not in DNA. Please remember, the name of nucleic acid is actually given based on sugar only, not based on nitrogenous base, based on the sugar. Ribose sugar containing nucleic acid, ribonucleic acid. Deoxyribose sugar containing nucleic acid, deoxyribonucleic acid. So name of sugar is very important. This is for the ribose. Then what are the deoxyribose? This is also like ribose structure only, but D means removal of oxygen, from which position? From the second carbon position. From the second position, originally OH is present. From this OH, remove O so that H will be present. Structurally, if you see, hydrogen will be there. But originally what happens? OH is removed, so by default hydrogen will come there. So OH removed, hydrogen coming means what is the overall difference? One oxygen is lesser. One oxygen less, oxygen is lost in the overall structure. So now what happens to this structure? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Last one will be CH2OH. Please observe this. As it is structure, except at second one. I am drawing this. CHO. This is OH. OH. H. 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 At the second carbon, observe carefully. In the original ribose, OH is present. In the 2-deoxyribose, H is present. That's it. This is called as 2-deoxyribose. This is the very important. If you know this, simply change this. Over. Okay, and this is the capital D at the second carbon loss of oxygen, right? So 2-deoxyribose. That's it. If you know this structure, that is the structure you are drawing. Okay, so this is the first carbon, third carbon, fourth, fifth. Understood or not? So this is the structure of 2-deoxyribose. Over. Completed. Very simple. If you know one, you can easily write the other one. Now come to this one. And here what we made, we were trying to make it as a ring structure. Try to do the same thing. See, 1, 2, 3, 4, 5. This is the CH2OH and this is the OH. And here instead of OH, H will be there. This is important for us. Okay. And here this ring will be connected here. H will be there. Here also left side OH is present. And here it will be H, H, H. Now what it will be? Left side OH. So that's why it is beta. And here right side OH. So that's why it will be capital D. And what is this one? At the second carbon, deoxy. So 2-deoxyribose. Very simple. If you know this, you can easily write that. Completely over. Very simple. So that's why try to understand the technical aspects so that it will be easier. Now the same structure we want to write here for 2-deoxyribose. Now here O and this structure will be like this. The problem is with which one? Second carbon only. So CH2OH write like this. And here above OH, below H, and second carbon leave it. Now here OH, H, and this one is H. Only at the second carbon, observe carefully. And here what happens? Instead of OH, H is required. So here H is there. That's it, over. So if you know the structure of this, you can easily write the structure of other one. So it is 2-deoxyribose. It is 2-deoxyribose. Beta, capital D, 2-deoxyribose. That's it. So in this way, we are studying about ribose and deoxyribose structures. Ribose is seen only in RNA and deoxyribose is seen only in DNA. So this is what about the nucleic acids and their corresponding sugars. With this, sugars part is also complete. And the last one is regarding the phosphate group. This is a very, very small information. Let us quickly go through this. Phosphate. Phosphate, it is also called as inorganic phosphate. Inorganic phosphate. Inorganic phosphate. In many reactions, you might have studied about this. You might have written as PI. Mainly, you might have studied about ATP hydrolyzed to ADP plus PI. What is this PI? This is the PI. Same PI. This is also. Okay. So anyhow, we will study about ATP, ADP, all the things in the next coming lectures. Don't worry. All these things were given in NCERT, so we will study all this. Phosphate, simply phosphate, or you can also call this as inorganic phosphate, or you can also call it as phosphoric acid. Phosphoric acid. In the previous lecture also we studied, no? Phosphoric acid. Or more clearly, you can also call it as orthophosphoric acid. Orthophosphoric acid. Please observe the spelling very carefully. It is a phosphoric acid. It is not phosphorus acid. Please observe this. It is phosphoric acid. Okay, what is this phosphoric acid? H3PO4 is called as phosphoric acid. While writing this PO4, H3, some students are writing P small O4. Please remember, this is actually capital P on the side capital O. Understood? That means it is phosphorus, this is oxygen. But if you are writing capital P small o, what happens? This will become polonium, right? Already you have studied about that table, right? So that elemental table, right? Periodic table. So capital P small o is polonium, but we don't want polonium. We want capital P capital O. Many students are making mistake here. So please observe each and every point very clearly. Don't make any mistake. Okay. So in this way, H3PO4. This is called phosphoric acid or orthophosphoric acid. From this, if H plus ion is removed, then what will be remaining? H2PO4 minus will be there. That is called as phosphate or inorganic phosphate. That's the same thing. If H plus ion is removed, 8 at the end, 8 you are using. If H plus is also present, you are saying that ic acid. That's it. That is the simple difference. Now we will see the structure of this. H3PO4. Now let us see for H3PO4, right? Yes. So here at the center P will be there. What is the valency for the P? It will be 5. 5 in this double bond O, and here it will be OH. This will be OH and this will be OH. Observe this. P, single P is there. Hydrogens 3, 1, 2, 3 hydrogens are there. Oxygens 1, 2, 3, 4 oxygens are there. This is how you are writing. Suppose the same thing if you are writing as H2PO4 minus plus H plus. H plus ion is removed. So from somewhere, from the OH, you can remove the H plus. So how you will write this one? P double bond O. This is OH, OH, and you can write it as O minus. Instead of OH, since H plus is removed, it will become O minus. So it is called as H2P1234 O4 minus. That's it. So this is called as phosphate or inorganic phosphate, and this is called as complete one, phosphoric acid or the phosphoric acid. That's it. So you can write this one or this one, anything you can write. But the important point is, see here. Because of this negative charge is obtained, negative charge. Please remember here, DNA is always negatively charged, RNA is always negatively charged because of phosphate group. This phosphate group here H plus is generally lost, so that it is getting the negative charge. So nucleic acids, whether DNA, RNA, whatever it is, always they are negatively charged molecules. That is the reason why actually there is a separate chapter called biotechnology. There we will study about movement of the DNA in between the electrodes. Suppose this is a unit and here you have given the plus electrode, minus electrode. Tell me the name of this plus electrode. Plus electrode means anode or cathode? Plus is anode, and the minus is cathode. Please don't confuse between anion and cation. Understood or not? Anode is having positive charge, but anion is having negative charge. What is the anion? It is the ion that is moving towards anode. So negative ion will move towards positive. So that's why anion will have negative charge, but anode is having, it is the electrode, right? It is having positive charge. Whereas cation is having the positive charge, so that it is moving towards the cathode, negatively charged electrode. Yes or no? So anode, cathode. Suppose we are taking DNA at the center. Suppose on which side it will move? DNA will move towards which side? Anode side or cathode side it will move. So what is the charge of DNA? It is negative. Same way for RNA also. Whether DNA, RNA, no difference. Both of them are negatively charged. DNA is negatively charged, RNA is negatively charged. So on which side it will go? It will move towards anode. This is the most important feature that you need to understand, and this logic is used in biotechnology. Okay, in that chapter separately we will study this. So please understand, DNA and RNA are negatively charged molecules, and how they are getting the negative charge. DNA and RNA are polymers of nucleotides. In every nucleotide, one phosphate is there, right? This is the basic unit of, in basic unit is the nucleotide. In every nucleotide, one phosphate is there. That means every nucleotide is contributing one negative charge. So in this way, this entire DNA, entire RNA, whether it is a small DNA fragment, large DNA fragment, whatever it is negatively charged. So always it is moving towards anode, that is positive charged electrode. These are the important points, and with this we have completed all the basic structures that are related to nucleotide. Nucleotide is having three components: nitrogenous base, sugar and phosphate. Nitrogenous base, two types: purine, pyrimidines, and then within the purine again adenine, guanine. Clear? Pyrimidines 3 types, cytosine, uracil, thymine. Sugars 2 types, ribose, deoxyribose. Clear? And then finally it is a phosphate group. And regarding this DNA, this one, deoxyribose sugar, and here we have written the molecular formula, right? C5H10O5. But here what will be the molecular formula? Carbons, number of carbons, there is no change. And what about hydrogens and oxygens? This is the last point we are completing. What about the hydrogens? Is there any change in the hydrogen? So OH, instead of OH, H is there. That means number of hydrogens remains same. So it will be H10 only. But what about the oxygens? When compared with this, here one oxygen is lesser. So this will be O4. Please observe this carefully. So deoxyribose is having C5H10O4 as a molecular formula. But this is having O5 as molecular formula. Regarding carbon and hydrogens, it is same in both of these. This is another small difference that you need to notice. That's it. So with this particular point is completed, and with this lecture is completed. In the next lecture, what we need to do, we need to combine this. Now we have studied individually nitrogenous base, sugar and phosphate. In the next lecture, we will try to combine this, so that we will try to write what are the various names they have mentioned in NCERT. So all those things we will discuss in the next lecture. Okay. Thank you so much, my dear friends. We will meet in the next lecture. Thank you.